In this segment, we will move on towards the various experiments regarding photosynthesis. So earlier we have discussed the three basic experiments which have proved the importance of carbon dioxide as well as sunlight for photosynthesis. Now we are starting with the role of atmospheric oxygen or air in the photosynthesis. That was Joseph Priestley experiment. Series of experiment he has performed 1770. So he has performed series of experiment using candle, burning candle, mouse as well as inverted bell jar. So he has performed series of experiments using mouse, burning candle and bell jar. Basically the bell jar that he has used is of inverted bell jar. So in the set A, what he has done is, within that bell jar he has placed the candle, and with the candle within that bell jar the mouse was also there, right? Mouse, M for mouse. So when the burning candle was exposed towards the mouse and the bell jar was placed, what he found is mouse dies. Why mouse dies? Because this burning candle produces carbon dioxide and he was not getting the sufficient oxygen because the burning candle was producing carbon dioxide and this jar was placed on the top of it. Candle also stops burning. In the set B, certain modification he has done. What he has done is, within that bell jar he has introduced the mint plant, right? So mint plant he has introduced, mint plant, candle also there and mouse also there, right? Now this setup was exposed to the light, plant received the light, performed the photosynthesis, candle was also burning, CO2 was produced by the candle, accepted by the plant, fixed by the plant, and in turn plant performed photosynthesis, releases oxygen. Oxygen is utilized by the mouse. Here mouse survive. So what is the indication that the oxygen is also important in the photosynthesis reaction? Right? So statement that he has given is, and this Priestley has discovered oxygen in 1774. Oxygen discovery, the credit goes to Joseph Priestley only. So statement that he has given is, plant restore to air whatever breathing animals and burning candle removes. So plant restores to the air whatever breathing candle and it is breathing animals, that is the mouse, and burning candle removes. Remove the oxygen and plant restores this oxygen, that is air. And in 1774 he has discovered the oxygen, that you have to remember. Based on the experiment of Joseph Priestley 1770, again one scientist, that is Jan Ingelhaus, used the same setup but with slight modification, right? He has used the aquatic plants and the bubble was produced. So experiment of Jan Ingelhaus experiment. So one more thing, Joseph Priestley experiment indicate or proves the role of O2 in photosynthesis, and the mechanism I already explained. Jan Ingelhaus uses or utilizes the same setup, right, but with slight modification, that he has used aquatic plants. So when the aquatic plant was used, the one setup he has exposed to light, one setup same was exposed to the dark. The setup which was exposed to light, he found that near the green parts of plant, that is aquatic plant, he found bubbles to be released out. And later on, these bubbles was identified as the bubbles of oxygen. Right? So again, this was the experiment that was performed by the Jan and Janhaus with slight modification. Modification is he has used aquatic plants. Now, sixth experiment is of Julius-Von-Sack experiment. In the Julius-Von-Sack experiment, based on this experiment, the statement that he has given is that green plants, let me write, Julius von Sack experiment of 1854. Green substance in the plant is located. Green substance in plants is located in the spherical bodies. Green substance in plants. Basically, now we know that this green substance is chlorophyll, is located in spherical bodies, and now we know that these spherical bodies are nothing but chloroplast. Synthesizes glucose, and this glucose is stored in the form of starch. So green substance in the plant, now we know it is a chlorophyll, is located in the spherical bodies, chloroplast. Synthesizes glucose, and this glucose is stored in the form of starch storage. Right? So this was the experiment. Based on his experiment, he has given his statement. Then seventh is the Engelmann experiment. In Engelmann experiment, he has used the green alga, that is Sclerophora alga. And one basic extra thing that he has done is, along with the culture of this algal species, he has used the culture of bacteria, basically aerobic bacteria. So he has used green alga, that is Sclerophora. These experiments are very, very important. Please do not neglect any of the experiment which I am discussing. So along with the green alga, he has used aerobic bacteria. The purpose of using aerobic bacteria, aerobic, which utilizes oxygen, basically is to detect sites of oxygen production. That means he wants to know where exactly this oxygen is produced. So what he has done is, he has taken the prism, or let us write first white light. He has exposed the white light to prism. We know when the white light is exposed to the prism, the 7 colors Vibgyor will be formed. Right? Vibgyor was exposed to the green alga, that was Sclerophora, and this is the mixture, alga plus bacteria. What he found is, bacteria tend to accumulate within the two regions of the spectrum, that was blue and green. So aerobic bacteria accumulate in blue and red regions of spectrum or spectra. So later on, it was also concluded that this blue and the red regions was the first absorption spectra of photosynthesis, and this absorption spectra corresponds to CHA, that means chlorophyll A as well as chlorophyll B. So wherever the bacterial accumulation takes place within the culture media, this was the site of oxygen production. Because wherever the oxygen is there, only at that place aerobic bacteria can grow. Name is aerobic only. Right? So aerobic bacteria utilizes oxygen for its growth. Then 8th experiment is of Van Niel experiment. He has used purple as well as green bacteria, green photosynthetic bacteria. Van Niel experiment. The statement, the conclusion was that photosynthesis is basically a light dependent reaction. So he has used purple and green bacteria and he concluded that PS, photosynthesis is a light dependent, it is a light dependent reaction, not bacteria, it is a light dependent reaction. Right? So this was the experiment that was performed by the Van Niel, and based on the experiment that he has performed using purple and green bacteria, he concluded that photosynthesis is a light governed or driven process. Hydrogen from suitable H2 from suitable oxidizable compound is used to reduce carbon dioxide. That is, we know now H2O plus CO2 will give you the glucose. Balancing we have to do for the reaction. So on all these things, one reaction that has been proposed, a kind of empirical reaction, empirical equation regarding photosynthesis was proposed. After all these reactions or all these experiments was CO2 plus H2O in presence of light produces CH2O